Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Nur Syafiq bin Muhammad Hakim. My metric number is J22B0043. Now I will present the introduction. As we know, biotechnology is the growing fast and needs system that can separate biological molecules and help enzyme works better. One useful tool is nanostructured membranes. These are very tiny filters with special surface and tiny holes that can be adjusted for different tasks. They work by letting certain molecules pass through while blocking others based on size, charge, or chemical makeup. Common types include microfiltration, ultrafiltration, nanofiltration, and reverse osmosis. Besides filtering, these membranes can hold enzymes in place, helping them last longer and work better. There are different methods to do this, like sticking enzymes to the surface or trapping them inside. This report looks at how these membranes are made, used, and how they can be improved for large-scale use in industry.
For fundamental concept of membrane technology, nanostructured membrane represent as advancement in modern biotechnology, specifically for protein separation and enzyme immobilization. And first is nanostructured membrane in biotechnology. Application is for protein separation, enzyme immobilization. Advantage is high selectivity, efficiency, and functional versatility. Fabrication is on nanoscale material, advanced method. Second is membrane separation principle. Mechanism is size exclusion, charge interaction, and chemical affinity. And next is pore size control, which is around 1 to 100 nanometer for targeted separation. And third is types of membrane process, which is first microfiltration, stand for MF, process around 0.1 to 10 micrometer. Second is ultrafiltration, which is UF, process around 2 to 100 nanometer, ideal for protein separation. And third is nanofiltration, NF, which is process around 2 nanometer, target small protein and ion. And lastly is reverse osmosis, which is RO, process around 0.3 to 0.6 nanometer, and it's suitable for applications such as water purification and biomolecule separation. And next is protein separation used by using nanostructured membrane. Selectivity based on size, charge, molecule weight. Example, an F membrane with hydrophilic layer, for example, carbon quantum dots, reject more than 90 percentage of certain amino acids. Benefit is high permeability, high flux, and large surface area. And lastly is enzyme immobilization technique, which is physical adsorption, a simple and reversible technique, and also has low stability. Second is entrapment, which is stable, enzyme in gel or matrix, and limited substrate access for it. And third is covalent attachment, which is strong and durable. And also it may reduce activity such as active site alteration. And lastly is bioconjugation or cross-linking, which is for site specific and stable. It often used in biosensor and bioreactor. And next for materials. First is biopolymer. The example materials for biopolymer is cellulose, starch, agarose, carrageenan, chitosan, and it features is water insoluble, strong gels, and it is easy chemical activation. And it also enzyme binding, which is reversible or irreversible and can be crossing for better stability. And second is nanostructure support. Um, it types there is various types of it, which is nanoporous material, electrospun fibers, and magnetic nanoparticles, and also nanotubes and nanosheets. Benefits from this material is it It provided high surface area, tunable size, enhanced enzyme and loading and efficiency. And third is inorganic nanomaterial. Example material is silica NPS, ZNS, Gitosan hybrid. And its characteristic is strong chemical or mechanical stability, and also it enhances thermal and pH tolerance. And lastly is hydrogel and cryogel. For example, PVA cryogel. It used in enzyme entrapment. The challenges is risks of enzyme leaching, requires cross-linking or compensation strategies.
Okay, now I will continue the part from Wani just said just now, based on materials on her part. Now I will continue my part, which is key design for consideration. The first one is surface chemistry modification. The second one is enzyme orientation and density. The third one is mechanical and thermal stability. The fourth one is the porosity and surface area. And number 5 is the magnetic properties. And number 6 is the microenvironment engineering. So the first one, I will talk about the surface chemistry. I have 2 points here. The first one is a functional groups, for example, aldehydes for covalent enzyme attachment. The second one is enhanced enzyme stability and retention. The next part is the enzyme orientation and density. The first point is a proper alignment maximize activity. The second point is the nanomaterial, shape effects, packing and interaction. Okay, we go into the next, mechanical and thermal stability. The first point is a cross linkers. For example, glutaraldehyde strengthen structure. The second one is improve durability of immobilized enzyme. Now we go into the next, porosity and surface area. The first point is the high porosity equals to the better enzyme loading and substrate diffusion. The second one is improved reaction efficiency. The next part is the magnetic properties. The first point is the magnetic NPS enable easy recovery and reuse. The second one is the boost operational stability. And lastly is the microenvironment engineering. The 2 points is states here creates optimal condition for enzyme function. And the second one is the extend enzyme lifespan and reduce post. Now I will pass to the next presenter.
So now I want to present about process description and mechanism. So the first process descriptions, protein separation using nanostructured membrane is achieved through a combination of physical and chemical instructions. The membrane contains nanopores that enable molecular filtration, allow the protein to be separated based on the size, chance, and specific binding properties. Such evolution commonly performed through utilization permits smaller protein to pass through the porous village one retard. Electrostatic interaction between a chance surface of the membrane and the protein also contribute to separation, as protein with opposite chance are attracted the return. Additionally, membrane can be functioned with specific ligand that selective bind to target proteins, enhancing the precision of the effection of the expression for complex biology mixture. So next, for the mechanism, protein separation rely on the virus mechanism to selectively isolate protein based on the physical and chemical property in size, elution, large protein, and retained by the membrane, while smaller protein pass through the porous, allowing for effective molecule filtration. Chance-based expression occur when protein are attracted of the repelled by the membrane surface depending on the solution pH and the protein isolate electric point affinity binding the features. In chance selectivity, as a specific ligand on the membrane surface bind only to target protein with matching recognition sites. Additionally, hyperophobic hydrophilic interactions, infuses suppression by screening protein based on the surface characterized, ensuring efficient and target isolation from complex mixtures.
Next, I will present about application. Nanostructured membranes are widely used in medical biosensors like those for diabetes monitoring and sports health. These membranes help enzymes stay active longer and perform better, which makes biosensor more faster and more accurate. According to Chen et al. 2020, graphene oxide-based membranes improve sensitivity and can detect very small amounts. They also allow a real-time tracking of body signals, which is especially useful in wearable devices for both personal and clinical use. Also, nanostructured membranes are important in the pharmaceutical industry, especially for separating proteins when making medicines like monoclonal antibodies used to treat cancers and autoimmune disease. These membranes remove contaminants while keeping the target protein safe and active. In processes like tangential flow filtration, they improve both the yield and purity of biosimilar drugs, as noted by Zhang et al. 2021. Compared to traditional methods, they are faster, cheaper, easier to scale up, and help keep proteins stable during large-scale drug production.
For case study, Zhao et al. 2019 studied DNA nanofiber membranes for separating a protein called BSA. The membrane's tiny fiber and connected pores gave it a large surface area, which made the separation process fast and efficient. It also allowed liquids to flow through easily and was more effective than traditional membranes. Even after several use, the membranes keep performing well, showing it's strong, stable, and suitable for protein purification. Nanostructured membranes offer great potential in biotechnology, but there are still key challenges and limitations holding them back. Technically, membranes fouling, difficulty separating similar proteins, and uneven enzyme loading can reduce their performance and efficiency. Material-wise, some enzyme or protein don't work well with certain membranes, and it's still hard to produce this membrane consistently on a large scale due to unreliable nanopores formation. There are also important limitations. Making membrane with precise nanoscale feature is both complex and expensive, limiting industrial use. They can become unstable and under extreme pH, temperature or pressure, and enzyme may detach, which lower the efficiency and reusability. Lastly, safety is a concern. Some nanomaterials may be toxic or build up in organisms, so more research and regulation are needed to ensure their safe use. Overcoming this issue is essential for fully using nanostructured membranes in biotechnology.
Assalamualaikum and hi, my name is Nur Atiasa Fiabita Adwazali. My metric number is J22A0290. My part is vaccine advice and innovative in nanostructured membranes. First, new material and membrane design. Do graphene oxide from surface stable for immobilization. Second, carbon nanotube coordinative unsafe in biosensor. Third, metal organic framework, highly porous and selective structure. Lastly, functional polymer can be modified for protein and enzyme selective. Next, to move on to cell-thermal specific membrane. In specific to pH, temperature, and light, control protein release and enzyme activity in a controlled manner, stable for automatic reaction system and biosensor. For innovation in nanostructure, firstly, 3D printing and microfluidics technology. 3D printing enables constant design of membrane, combined with microfluidic chip for small-scale application, then stable for lab on a chip system and Genox steel. Lastly, bio-inspired design and hybrid membranes, mimicking biological systems such as aquaporin for ultra-selective fusion, hybrid membrane, a combination of organic and inorganic material, and add nanoparticle TRO and ZNO for anti-foliation and height strength. For visual application, biomaterial, food process, continuum bioreactors, and biosensors. Thank you. That's all for my part.
So last is conclusions. Nanostructure membrane enable molecular suppression and enzyme immobilization while high infection that, thanks to the large surface area and adjustable pore size, is a high selectivity for virus applications such as protein separation, biosensor, and bioreactor. Performance is improved through simulated responses, material height composite, and bio-inspiration design, predict to be the main key in sustainable and high-capacity biotechnology system in the future. Thank you.